Go through today and get started, and then that'll lead into also some descriptions of your assignments for next week then as well. So we're going to talk a little bit about the lab today, but we're also going to talk a little bit about papers, again, the scientific papers in general, but more specifically the discussion sections of papers as well. Okay, so first off, before we get started, just a reminder, there were 3 assignments due today, just like there has been almost every week. There was the pre-lab worksheet over today's lab. That was actually a 10-point pre-lab as well, as opposed to the normal 5. That is because this week's lab is the first part of a 2 weeks long lab. So the stuff that we're going to do this week and next are very related to one another. But I just gave you 1 pre-lab, so you didn't have to worry about another one next week. So we're going to do that. That pre-lab is due today, but remember, it was 10 points. So if you haven't finished that, you want to finish it because it is worth a bit more than a normal one. You had the communication worksheet number 4, and that was the last one. We're done with those now, but make sure if you haven't done that, do that. It does help cover discussion sections, which I'm going to talk more about here in a second. And then we also have the methods assignment, and the figure that went along with it was due today by the beginning of lab. If you haven't gotten that in, please get that in as soon as possible as well. With these writing assignments, I sent out an email to the class. Always have been sending out email reminders I emailed on Wednesdays about the assignments that are due the next week. I emailed on Mondays about the assignments that are due the following day. But I did just want to point out there is, if you haven't gotten into these writing assignments, it's really important for you to get them in by the 17th or 18th at the absolute latest of October. And I'll talk a little bit about this one, but we are coming up on to their first full paper assignments are going to be due October 25th. But these smaller writing assignments don't take too long to do, and they're meant to help you prepare for that full paper assignment, where it is basically the version of an exam in this particular course. But because the first full paper is due on the 25th of October, I really need to be seeing your individual smaller writing assignments before then, so that I can grade them, so you can see how you did, so I can get feedback on you, not on you, okay, to you. That's weird. All right. And so I can get feedback to you so that you guys can do better on those future writing assignments. So I really, again, my goal is not necessarily you guys are getting stuff, all these turned in, but you do have to be completing the assignments to pass this course. You do have to be doing these assignments in a timely manner. You got to be paying attention to your email, because I'm trying to be as clear as possible as when everything is due. And if you're not getting that stuff in on time, it does mean that's when I end up failing students in the class in the past. It's not very common, but it's the one thing. I'm going to try to make it easy as possible with many reminders, but you guys have to sit down and do the work as well. And I've got to do it at enough time for me to give you feedback so you do well in the course too. So just a quick reminder about that one. So I am wanting to see those if you haven't got them in soon. Today, though, we're going to talk a little bit about just abstracts today. We'll talk about discussion sections today, papers in general. We'll be talking a little bit about what are enzymes, why are enzymes biologically important, what affects enzymatic activities. And this is going to be the first part of this 2-week lab, what we're going to be doing this week and next week in lab. First, again, the paper assignment, it is coming up. It's October already. The semester is kind of flying by. And so we even got things like fall break coming up here after. So we have lab class on the 11th next week, and then you guys get fall break. But that also means that first full paper assignment comes up much quicker than you think it's going to as well. The first full paper is over today's lab and next week's lab, both. So both the labs, and what we're taking a look is the enzymatic activity of a particular enzyme called catalase. And by the way, if you ever see anything with the ASE suffix ending, it always means enzyme. ASE just means enzyme when we're talking about biological terms. But we're going to look at catalase's ability basically to break down hydrogen peroxide. How many of you have ever used hydrogen peroxide on a cut before? Let's start doing it. It buzzes up and it makes bubbles, right? Those are basically catalase. We're going to talk about this later. But when catalase, what it does is it breaks down hydrogen peroxide. And when it breaks it down into water, H2O, and O2, oxygen gas. And what you're seeing is oxygen bubbles there. And you're going to see that in today's lab as well. And they have the catalase. That's because we have the catalase enzyme inside of our bodies, inside of our cells, to detoxify hydrogen peroxide. A lot of bacteria have catalase enzyme. Yeast that you guys are going to be working with today has catalase enzyme. Okay. The paper, though, is going to be over. We're going to use catalase as the enzyme we're investigating. But really what we're trying to focus on is how is the concentration of enzyme, how does that influence enzymatic reaction rates? How does temperature influence enzymatic reaction rates? How does pH influence enzymatic reaction rates? Chemical reaction rates, how are those influenced by those environmental types of conditions and by enzymes? And we're just tracking, this is going to be for enzymes in general, but we're tracking catalase enzyme more specifically. The paper is going to be due again on the 25th. It is worth 75 points. It's also a required assignment for this course. There's 2 full papers in this class. Failure to complete any one of them is an automatic F, failure for the course. So they have to be turned in and they have to be done. Because that is the main objective of this course. One of the main objectives, again, is yes, investigating cellular and molecular biology with our labs, but also the writing assignments and scientific writing. So paper assignments, again, we talked a little bit about this. They go to the title page, abstracts, intro, methods, results, discussion, literature cited in that particular order. We've been practicing writing some of these. So far, you guys turned into methods today. We had an intro last week. We had a results section the week before that. We had a literature cited sections at the beginning of the semester then as well. So we've been practicing writing some of these individual writing sections over time. I do recommend always writing the methods and results first. The methods is the easiest section to write. The results is, you have to write this before you can write the intro to discussion. You have to know what your results are. You have to kind of figure out what your overall narrative storyline for your paper is going to be. There is a little bit of creativity to scientific writing because you have to have a story, but you don't know what your story is until you know what your data is saying to you. Does that make sense already? Okay. Then you typically write some combination of the discussion, intro, and let's cite it together. And then finally, you end up with the title and abstract at the end. Can't write the abstract at the beginning because it's a summary of the rest of the paper. If you want to summarize the rest of the paper, you kind of need the rest of the paper first. Again, I talked about this earlier, but you should be outlining main concepts. Don't just start writing stream of consciousness like my 5-year-old. Or not my 5th grader, sorry, is what I was going to say. He's been working on a book report for the last week or so, and it was just like all over the place. You need to basically sit down and think what your story is going to be. What are the points you think you have to make? What is the important stuff that should be in this paper, and what order should it be in? After you got an outline, then you can write your first draft. I always recommend starting early so that you can take a break. A lot of times I'll put something on a page and I think I'm being clear in my communication, but I'm not. And so I know what I was trying to say when I was writing it. It's not necessarily what I actually put down on the paper. So, and then if I read it through, sometimes my brain still knows what I was trying to say, and it fills in missing information that wasn't actually there in the first place. It happens all the time. So take a break, revise the draft. And again, I'm always available for, you can get feedback from Ace on your writing. You can get feedback from me on your writing. So as long as you guys are getting an early start, I'm more than happy to take a look at drafts of your paper or even drafts of individual sections out of your paper as you guys are working on them. And so I'm more than happy to take a look at those and offer any feedback and help out. Because again, that's just part of the process. I want you guys to be seeking feedback. That's what all scientists do when they're writing a paper. When I've written papers in the past, I'm usually sending it out to 4 or 5 other people just for them to do a read through and offer feedback on what needs to be improved as well. Okay, and then you revise it again based upon that feedback. But again, that's the type of thing you can't wait to the last moment, can't wait to the night before. Otherwise, you end up like my wife, who was in college, and she hates me telling this story, where she was doing an English creative writing class, and she got done writing it, and I'm like, yeah, you just wrote To Kill a Mockingbird. She's like, what? No. Oh, no. She literally basically wrote, didn't rewrite Kill a Mockingbird, but it was the same kind of general family unit and not the same exact situation. But I'm just like, yeah, that's To Kill a Mockingbird. She had to turn it in and her professor didn't catch it. Anyways, so, but she was very upset because she did not mean to do that. Discussions, that's one of the sections we haven't talked about yet. So we've talked about, for instance, abstracts in the past. We've practiced intros, methods, results now. Discussion section is actually the first time you turn in a discussion section will be on the first full paper. So you guys are not going to get a practice run at the discussion section. So the first one will be on the first full paper. So I do want to talk about this. Discussion sections are for drawing conclusions, interpreting the data, and explaining the data. In the results section, you describe the data itself. Very straightforward, be very detailed. You describe trends or patterns in the data that you're being observed. Like for instance, earlier this semester when ocean water was much, much higher than any of the others, that's an important trend and should be described in the results section. The discussion section is more about interpreting and explaining the why. Why did we see this trend? What are some possible reasons that this occurred? So that's what we're really talking about with conclusions, the why and interpretations of the data set. Typically though, you have to do this as a form of communication. You still have to keep in mind, you're trying to communicate to your reader and you want your reader to follow along easily. So typically you need to restate your purpose, restate your hypothesis that you have or hypotheses if there's multiple ones. Do not ever expect your reader to remember this all the way through. Don't expect your reader to do your work for you. Instead, you got to be super clear. Purpose, restate a hypothesis, do it explicitly, clearly. Then you often are going to tie the conclusions to your data. But this typically means, again, briefly summarizing the data. You don't have to go as in much detail as the results section, but you do need to say what are some of the general trends or patterns that you saw. So briefly summarize the data. And again, this is to help your reader follow along. And then once you've summarized the data, now your readers where they need to be, they're all caught up. And then you go into your interpretations. So you interpret your results or trends. You explain why the data is the way the data is. What are some reasons for this? You can talk about, is your hypothesis supported or not? But also you should be talking about why is this important? So it's a big set of whys. Why did this happen? Why do we see this trend? Why is it important? That's what you're focusing in on the discussion section. And again, it's hard for me to give some specific examples because the why is going to be different for different experiments. You're going to have different explanations for different kinds of experiments then. But this is what you're going to try to focus in on the discussion section. The important thing is also, just like in the results section, you still have to cite figures parenthetically relevant. So for instance, in that little part where you're summarizing your data, if that data is contained within a specific figure, you want to make sure the reader knows where it's at. So you cite it by doing a parenthetical citation. So if this could be my sentence where I'm summarizing the data, and then I do open parentheses, figure 1, close parentheses, period. And that's my parenthetical figure citation as well. So you're still doing that in the discussion section, just like you were doing that in the results section. A lot of times what I'm also recommending is that you are doing all of this, but in a paragraph, and then there's about one paragraph per figure is the way this is generally going to work for this particular class. So for instance, again, in the paper assignment I said we're going to do today, there was a concentration portion, there was a temperature portion, and a pH portion. Well, guess what? That's 3 different figures. And for your paper, then, if there's 3 different figures, and I'm saying there's a paragraph per figure, that's at least 3 paragraphs in your discussion section as well. One, where you're doing this entire process for the concentration portion of your experiment. Another paragraph where you're doing all of this again, but for your temperature section. And a third paragraph where you're doing all of this again, purpose, summarize the data, state the hypothesis, interpret it. And you're doing that for the pH portion of the data. Questions about that? Okay. The other things you sometimes hit in the discussion section, and this is again involved in interpretation and explanation, if needed, if you think there was a possible problem with your data, you have to bring that up. You can't sweep it under the rug and cover it up. Problems in data sets have to be addressed. So if needed, if there was any potential problems in your experiment, things that didn't work right, data looks wonky, wasn't what you expected. Okay, if those should be mentioned, and if there were mistakes, how can those mistakes be fixed? How could the study be improved if you repeated it, for instance? Okay, so that's what's your other thing that should be part of the discussion. Again, though, I have the if needed. If everything seems like it's worked, that's okay. You don't have to make up problems where there were no problems. The other thing that you have to do in the discussion section is literature citations. Have to. So literature citations are a required part of discussion sections. Again, your main conclusions should be drawn directly from your experimental data. That's your main conclusions. And that's part of what I talked about previously. However, this is also a section to compare your work against other research that has been done, other literature sources. So this is also where you want to do some of those types of comparisons. And typically what you're doing is you are either citing some paper, a rather research experiment that is similar to your own and has found similar conclusions to you. So if you have these conclusions, this other research group did a similar experiment and found similar things, those support one another. It strengthens your paper and your conclusions. And that's usually what you do in this discussion section is you make some conclusions from your data set and you say, oh, and this other group also found similar things. Great, that's good. Occasionally, though, you might find papers that contradict your study. Although you gotta be a little careful. Did it contradict it because you're looking at human enzymes versus yeast enzymes, which may not behave exactly the same? An enzyme from a radish doesn't always behave the same way as an enzyme from a yeast fungus, for instance. That's a plant versus fungus enzyme. So you gotta be a little careful about that sometimes. But if you have any papers that contradict, then you want to address that in this particular section as well. Okay. Questions about that? All right, any sources should be used to tie and furthering your own conclusions. So they should integrate with the conclusions that you're making in your papers then as well. All right, so this is just kind of a slide that has a lot of what I just went through. You guys can read this real quick. I'm gonna get these started, put in an incubator. These are the yeast we're gonna work with. I'm just getting them started. But I'll be right back once I just gotta put it in the incubator real quick. Read through that one and we'll see if you guys have any questions. So again, this is just my little slide that's just ending. Oh, sorry, no, this wasn't the summary yet. Whoops. All right, well, I'm bad. All right, ending the discussion. The other thing is there should be a fourth, often and final paragraph to your discussion. And it kind of just summarizes the entirety of the discussion section then. So you end by summarizing, you end up with a final paragraph that summarizes all your main conclusions that you made. but also really what's the significance of your experiment and conclusions? Why is it biologically relevant? What's important from a biological standpoint that you found? Does your experiment have implication towards some type of application, for instance? And just why in general is important. You always kind of want to end the paper. This is the end of your paper. So you always want to kind of end on a high note as well. That's the way that that's just good communication with your reader too. Questions then? So again, just summarizing, it's usually one long paragraph per figure, and each paragraph should state your purpose / objective, summarize important trends in your data, interpret, provide possible explanations for the trends that you observe. Those are your conclusions, and compare your findings against other outside references, other experiments that have been done. And you're doing that in general, one figure per paragraph that's hitting all of that for each paragraph. And then a final paragraph that summarizes your main conclusions, discusses why they're important or towards biologically or towards some type of application. Questions about that? It means you also got to get started early and you got to do literature sources. And also the types of literature sources you need are typically in this section, not your textbook and not the lab manual. Those are typically not good sources for a discussion section. You really need to be looking for primary or secondary articles. And again, primary articles are first-hand accounts of research. That means experiments are done. The people that did the research wrote the paper and published it in a professional journal. Secondary articles are typically review articles. They're written by an expert in the field, but they summarize several different primary articles then into one spot. So those are usually the types of references you have to use for a discussion section. All right, questions about discussion sections. All right, today's lab is going to be over enzymes. What's an enzyme? It is usually a protein, but not always a protein. It's almost always protein, but not always. And not all proteins are enzymes either. So what's an enzyme, or what does an enzyme do? Jacob.
Speeds up chemical reactions.
So it's something that speeds up chemical reactions. However, it's a little bit more detailed than that. And actually, something that speeds up chemical reactions is a catalyst. Not to be confused with catalase, by the way. Don't get catalyst, which is anything that speeds up chemical reactions, with catalase. Catalase is the specific enzyme we're going to take a look at today. But an enzyme is a more specific version of a catalyst. An enzyme is a biologically produced catalyst, or basically a way of stating that is it speeds up chemical reactions. Usually it's a protein, but occasionally there are some RNA molecules that can function as enzymes as well. There are some enzymatic RNAs. So when I say enzymes is a biologically produced catalyst, catalysts can be things like just chemicals. They can be things like sometimes even temperature, for instance, can act as a catalyst. It's not something biological. An enzyme is a biologically produced, it's a catalyst that's made by a biological organism. And it's almost always a protein, but occasionally it is an RNA molecule. For instance, proteins are actually made by the enzymatic activity of a 23S rRNA molecule. So it turns out when you join amino acids into a protein, that is catalyzed by an RNA catalyst. Okay. And so there are a few select examples where RNA molecules act as enzymes. All right, enzymes again are biologically produced catalysts, but again, catalase are anything that speeds up a chemical reaction. So we're going to take a look at enzymes in today's lab and next week's lab. Then what they do is they speed up chemical reactions. So for instance, I might have my chemical reaction here. A plus B yields C and D. Which one are my substrates / reactants in the chemical reaction here? A and B is going to yield to my products, which are C and D. And that's just could be a generic one here. The thing to keep in mind is what is going to happen is this reaction, A plus B might come together, goes through a chemical reaction, makes C and D. But that takes a certain amount of time. And a lot of chemical reactions by themselves are pretty slow. And so a lot of times cells need to speed up chemical reactions a lot faster. And the reason is just chemical reactions are pretty slow. And they're so slow, they don't take place fast enough for a cell's needs, for an organism's needs. So they got to speed them up in some way. And what the job of enzymes are is they speed up these chemical reactions, but they don't speed up chemical reactions in general. You need a specific enzyme for a specific reaction. Enzymes are what are called substrate specific. They only bind to very specific substrates or reactants and help with that chemical reaction to the products, but that's the only reaction they speed up. So enzymes are very, very specific, and I'll talk about why that is here in a second. That being said, they do a really good job of accelerating chemical reactions. Enzymes will accelerate reactions by 10 to the 8th up to 10 to the 20th times faster than in the absence of an enzyme. So enzymes really speed up chemical reaction rates really quick. And enzymes are the dominant factor in controlling reactions inside of cells. How do they do this? They do this by lowering something called activation energy. So what I have here is a graph. Have you guys gone through this in lecture yet at all? No? Okay, that's okay. So what I have here is a reaction. And you might have reactants going through a chemical reaction over time, the progress of the reaction, and they give rise to some products on my little graph here. This is just progress of the reaction on the x-axis. This is then free energy, the energy associated with things and over the course of this reaction. From little energy to more energy at the top. And you might start off with the reactants. This might be the energetic state here of your reactants. But you can see as those reactants come together, as they go over the course of the reaction to form the products, you see their energy state changes in this particular case. In this case, the reactants start out of a higher energy state and the products end up at a low energy state. Does that kind of make sense to everybody? This is an example of something called an exergonic chemical reaction. And so that's what we're kind of looking at here. Now, if you look at this, though, without an enzyme, this is the course of the reaction without an enzyme. As the starting energy state of the reactants the same between without or with an enzyme, does the reactant energy state start at the same level? Yes. Does the products end up at the same energy level? Yes. So your starting point and your ending point is exactly the same, whether or not you have an enzyme in red or don't have an enzyme in black. What's different is the middle. And the middle is things like reactants coming together. It's things like the reactants getting close enough to touch and do things. It's things like breaking apart the reactants to reform new products.
You're saying the middle, you mean like how like close the way it is?
Yeah, okay. This is the middle. And what ends up happening, as you can see, is this line without an enzyme goes higher. But really, remember, as higher is more energy. So without an enzyme, you have to put in some energy to get the reaction started. Now, as you go over the reaction, you get some of that energy back, in essence, but you still have to put in a pretty big amount of energy to get this reaction started. This is called an activation energy barrier. And it is a barrier. The more activation energy you need to get a reaction going, the slower that reaction also goes, because that's a barrier to it taking place. But with an enzyme, enzymes lower that activation energy that's needed And because you need less activation energy, that reaction will happen more often and therefore more quickly. Does that make sense, everybody? So that's what we're really looking at here. So here we have, this is again, the activation energy, E sub A, activation energy. Without an enzyme, you need more of it. With an enzyme, you need less of it, and therefore there's less of a barrier and the reaction happens more quickly. Everybody okay with that? Sorry, Gio, you good? All right, how do they lower activation energy? Binding substrates to increase their local substrate concentration. This is a fancy way of saying they bring reactants close together. They help to ensure that they're oriented properly to interact, and I'll show you what I mean by this in a second. And then they also strain reactants. They put electrical or physical stress on the reactant, and that makes it easier to disrupt the reactants, get the reactants kind of come apart a little bit so that they get rearranged into the products. All right, so what does this kind of look like? Here is, for instance, my same similar chart. Progress of the reaction on the x-axis, free energy over here. But let's just say I have a couple reactant substrates. Reactants and the substrates and the enzymes, they fit together almost like puzzle pieces or keys and locks. They fit together just right. So typically somewhere in the enzymes, they have what is called an active site. And you can kind of see it's shaped to fit the substrates. You guys see that? It'll bind onto one substrate and hold on to it. Then it'll bind onto the other reactant or substrate and it holds on to it, but it also brings them very close together. When they come together, they literally are brought together inside that active site of the enzyme, and they're brought really, really close. And by binding onto them and holding onto them, it increases the local substrate concentration. It brings them together so close at a molecular level, they react. And you can form the products. The other thing it does when it does this is it orients them in the right way. So if you have a sugar groups, so say I have a say this is my glucose. It typically has a hydroxyl group coming off of here. If I have another glucose and I want to build and join them together, typically you build glycosidic bonds between sugars when this hydroxyl group reacts with the hydroxyl group on the other sugar. But are my hydroxyl groups oriented properly here? Can they bump into one another to go under a chemical reaction? They're on the wrong side. This one's over here, but this one's over here. The rest of the molecule is blocking things. Does that make sense already? But if I orient them properly, now I can bring them close enough together if they're oriented properly to undergo a chemical reaction. So that's what we're looking at with some of these. Okay. So not only do they bring them close together to do a chemical reaction, they also orient them properly to do the chemical reaction. That speeds it up quite a bit. By the way, you guys have probably heard that temperature, higher temperature speeds up chemical reactions. Why? What is temperature? Things have a higher temperature. What does that mean about them? Or what does temperature measure? What happens if you do this? Moving, I'm generating friction, which is giving me what? Heat. Heat energy. But why do things warm up? Because I'm moving them around and I'm rubbing them against one another, but they're moving around as well. What is heat? What is temperature? Temperature is a measurement of the heat within a sample. Heat is a measure of kinetic energy or molecular movement. Things with a higher temperature have more heat energy associated with them, and they're moving around more, even at a molecular motion movement. If you have reactants sitting in a solution and you heat that solution up, they start to move around more. So if that's cold, they're kind of just moving real slow. But if you heat the solution up, heat the water up, boil it, maybe they start moving around more. And then what do they do more of? If they're moving around more, they bump into one another more. And if they're bumping into one another more, they react more. Does that kind of make sense? Does that kind of sequence make sense? Temperature can speed up chemical reactions, but no matter how much temperature you put in there, it doesn't speed up chemical reactions a ton sometimes. But that is an example of activation energy. As you heat it up, causing the molecules to move more, they bump into one another more, they react more. But enzymes are way better at this than just increasing the temperature. Enzymes will increase rate. They bring together the substrates much more effectively than just raising up the temperature of a solution. The other thing that enzymes do is, again, you have that substrate fits into an active site in here. So my enzyme is over here. My substrate comes in, it binds to it. And then do you guys see what happens to my enzyme? Is it the exact same shape prior to the substrate binding? Here's before substrate binding and here's after. Did anything happen to my enzyme in blue? It closed and wrapped around it. And what it does is it squeezes it. It takes that substrate, that reactant, and it squeezes it. This is called induced fit. So substrate comes in, binds, reaction wraps, or enzyme wraps around it and squeezes it. This puts physical or electrical stress on the substrate. It starts to stretch the substrate. It starts to strain the bonds within the substrate. That makes the bonds easier to break, and you form the new bonding patterns of the products. So this induced fit model also explains why, because you're doing this physical electrical strain, it puts strain on the substrate, makes it easier to break it apart to reform the now formed products. That also lowers the activation energy necessary as well. Questions all about all that? The other thing to keep in mind with enzymes is they get recycled. So I might have an enzyme, has an available active site here, starting in my little diagram. Substrates come in, they bind into that active site, they fit together like puzzle pieces. You'll see my enzyme changes shape. There's that induced fit and it strains the substrates. And then they get converted into the products. Those products get released by the enzyme once they're formed, but now the enzyme is going to flip back to its original shape, where it started at the very beginning, and it's available to bind new versions of the substrates. And the enzyme can keep binding substrates once, twice, 3 times, 4 times. It keeps speeding up the exact same chemical reactions multiple times in a row. The enzymes are recycled during speeding up a chemical reaction, and then they're reused. They don't just speed up the chemical reaction one time, but multiple times over. Questions about that? So we're going to look at enzymes and how they affect chemical reaction rates. We're going to do that this week and next week. We are, though, again, I've already brought this up. What enzyme are we going to assay today? What's the name of the enzyme that we're specifically going to look at? Catalase. What is the source, though, of enzyme? Where are we going to get this catalase enzyme from? Am I just going to give you like purified catalase and then you work with it? Or am I giving you something else as a source of catalase? Yeast. So this is the enzyme. The source is going to be yeast cells, which is what I basically mixed into those flasks just a little bit a moment ago. I'm waking the yeast up right now. Anybody ever made bread? It's just baker's yeast. I'm activating the yeast. It's literally store-bought dry yeast from the store that you would use to make bread. Okay, that's what we're going to be working with. So we're going to be doing that one. We're just working with Saccharomyces cerevisiae today. This is a single-celled unicellular fungal organism. So it's a fungus. And it's one of the simplest eukaryotes. It's also one of the most studied eukaryotic cells for a couple different reasons. It turns out, though, a lot of the processes that go in yeast are very similar to the processes that go on in our cells. So for instance, most of what we know about from mitosis, which you guys will talk about later in the semester, is from studying yeast. And then we found out, oh, all these genes that are in yeast that are involved in mitosis, all these proteins involved in mitosis yeast, human cells got the exact same thing. There's a slight change here, slight change there, almost the exact same process though. And yeast, why research this? Really fast, easy, cheap to grow yeast. Not fast, cheap, easy to grow humans, for instance. Ask your parents, no. They have a really fast growth rate. So we can grow a culture of yeast in just a few hours to a day. So really fast, easy to grow. They have a fully sequenced genome. We know all of their DNA sequences. We actually know what a good number of the genes encoded in their DNA also do. We don't know all of them by any way, shape, or means, but we know a good number of them. And they're easy to manipulate genetically. I can delete a gene here. I can add a gene there. I can mutate this gene and change its sequence, and that is done all the time in research labs. And no, it typically just makes a worse version of yeast, not a better one. They are actually, because they're eukaryotic, they are a good approximation of human cells for certain processes, especially like mitosis. Mitosis in yeast is almost identical to mitosis in human cells, for instance. So we study the cheap organism, the one that's easy to take a look at. That lets us get a base, a foundation, and then we can extrapolate into more complex or harder to research organisms as well, or things like humans where there's ethical constraints in doing experimentation then as well. Okay. And most of what you guys hear about, for instance, if you're going through mitosis in your textbooks, it's probably the yeast version of mitosis that's really being discussed there. Yeast is also important for commercial uses. For instance, we use yeast to make bread and donuts and beer and wine and alcohol and a number of other commercial purposes then too. So with yeast, we're looking at these guys. We're going to take a look at their ability to do certain things. You know what? Yeah, I don't need to go through that. Specifically, though, I'm gonna go through that in a couple weeks. Actually, I'll come back to that slide. Sorry about this. I forgot to take them out. Okay. Specifically, we're going to look at catalase enzyme for yeast. Catalase. This is an enzyme that speeds up this chemical reaction. 2 molecules of hydrogen peroxide are then going to get broken down into 2 water molecules and oxygen gas. And again, if you've ever put hydrogen peroxide on a cut, the bubbles you're seeing are oxygen gas as hydrogen peroxide gets broken down. So that's what we're taking a look at. And you guys are going to see them bubbling and giving off oxygen gas in lab today. Hydrogen peroxide is a very common byproduct of cellular respiration. Our cells do cellular respiration. Yeast do cellular respiration. So hydrogen peroxide is a toxic byproduct, waste product of cellular respiration. But because it's toxic to the cells, they need to get rid of it. And they use catalase enzyme to detoxify this and get rid of it to water and oxygen gas, which is less toxic. So this is something yeast have to kind of protect themselves from their own metabolism, their cellular respiration waste products. We want to use yeast as a source of catalase. We're going to use catalase to then figure out how different types of conditions like enzyme concentration, temperature, and pH affect chemical reaction rates then. With this, how could you possibly measure the rate of catalase activity? What do you probably have to look at? If you want to measure the rate of reaction, what's one thing we could measure? We have to look at something being released, and we have to look at how it changes over time or rates. So if we want to measure chemical reaction rates, we have to look at the appearance of a product over time, or alternatively, we can look at the disappearance of a reactant over time. So the 2 ways we could track this reaction rate is we look at disappearance of hydrogen peroxide. That could be one possible way we do this. But we could also look for appearance over time, increases in the products. What are we actually looking for today, though?
Appearance of oxygen.
Yeah, we're going to be looking for oxygen gas Hence why you have the sensors out there that are labeled oxygen gas sensors with the labels on them in front of you. Okay, so those are what we're going to be looking at, oxygen appearance over time, but we're going to see O2 concentrations increase over time, and we're going to get rates. So specifically, we're going to basically be assessing chemical reaction rates. What we're going to be doing today, effective enzyme concentration and how that affects enzyme reaction rates. Next week, we're going to be doing temperature and its effect on reaction rates, and pH and its impact on reaction rates. This is important because, again, biological organisms have to have enzymes. Biological cells cannot survive without their enzymes. The chemical reactions simply by themselves do not take place fast enough to maintain a cell. A cell has to have these enzymes to process energy fast enough to maintain itself, to make structures it needs fast enough to make itself, or the cell would literally fall apart. So in understanding how enzymes work and proper enzyme function is really important for understanding how biological organisms and are successful and can live. And that's why, for instance, if you ever run a fever, you start to feel bad. You guys ever raise your body temperature? Your enzymes start not working as well. And that actually means you're not processing energy as well. That means your body's functions are lowered. And that's part of the reason they feel bad with fever. The thing is, your body makes you have a fever when you're sick to also do the same thing. It's trying to get rid of the enzymes and any pathogens in your body and lower their function as well. So the fever is actually an immunological response to try to help fight off the pathogen. It's hoping it's going to affect the pathogen, the disease-causing microorganism, more than your own cells. But questions about that? Okay. All right, quick protocol for today, almost done. And then we'll get started. You're going to get 3 test tubes. Go ahead and label them up. Test tube 1, 2, and 3. You're going to place 10 mils of the 1.5% hydrogen peroxide solutions out there. So you see the bottles that are labeled for the 1.5% hydrogen peroxide solution. Those are where you're going to, there's little tiny graduated cylinders out there. You're going to measure out the 10 mils out of those, and then you're going to pour the 10 mils into the plastic Nalgene bottles that are on the, I think, ring stands at the moment. Okay. You're gonna then, I'm going to get the yeast out here in a second. You're going to come up to the front and get a yeast solution for me. For the yeast solution, every time, but right before you use it, make sure you mix it really well. I think you guys have little bits of paraffin? No. You guys have little paraffin bits out there? You should.
No.
Well, that's fun. Okay. Yes, you do. In the finger bowls. Yep. Okay. Might have to share them a little bit. You're going to put a piece of paraffin over the top of the yeast and really shake it. Use the paraffin to seal it. You can actually just use your thumb if you want to, but the paraffin gives you a little tight. It's yeast. You eat it all the time. It's fine. But you give it a quick mix. That's because yeast are pretty heavy. And if you don't mix it, they all settle down to the bottom of the tube and you don't sample enough or consistently sample them. You get more in one of the tubes and less in some of the others. So the first time you're going to take the 10 mils, you're going to have it in the Nalgene bottle. You're going to add 5 drops of yeast. And then you're going to put that into you're going to put the oxygen sensor into the Nalgene bottle. You don't have to, you want to give it a gentle press, not very much, just a little bit. Actually, if somebody's using that one that had the previous group left their oxygen sensor out, you might want to take that out here. I might handle that in a second. But you're going to put it in just gently. Make sure there's a good seal. You're not pressing hard. You just got to give it wedge it in there just a little bit. Okay. Then you're going to collect data for 3 minutes, and what you're tracking is oxygen being produced by this chemical reaction over time. You're going to calculate rates. I'm going to let you not use Excel today. If you guys have been using Excel this entire time, it turns out there's a completely hidden way to actually just use Logger Pro with a click of a button and calculate rates. Why did I make you use Excel? Because you won't always have Logger Pro available, but you always have Excel available. So you need to know how to do this in Excel as well. Okay. So you're going to wash it out, dry it, and repeat it. But then again, a new 10 mils of hydrogen peroxide, but now with 10 drops of yeast added in. And then a third time, new 10 mils of hydrogen peroxide, but with a 20 drops of yeast added in. And for the drops of yeast, you're literally just using these guys. These are plastic pasteur pipette things. You're just going to press the bulb, go into the yeast, suck it up. You're then going to literally try to do drips. You're going to press it until count the drips pretty slowly. 1, 2, 3, 4. Don't go too fast, otherwise you're going to get an inaccurate measurement. And you're going to let the drops fully drip on their own. So don't lean it against the side, because then you can't see the drip form, or then you won't get a nice drip. You want to let the drips free drop in one at a time and count them, so that you're making sure they're being pretty accurate. Turns out a drip is approximately 100 microliters out of these. They're set up to be specifically that type of volume. Okay. And then you're going to determine the rates. Questions about that? All right, last thing I want to talk about is assignments for next week. No pre-lab, no communication worksheet. So no bright space worksheet quiz things. You do have another writing assignment. You have abstract over last week's lab, not this week's lab. So it's over the cell size agar cube lab. And this is going to be, again, I'll email this out after class, but it will be just an abstract section. And again, that's a summary of introductory material, method material, result material, and conclusions material in 250 words or less over the data you collected last week. Okay. So that's what this particular assignment is. I am also going to say that I expect at least a minimum of probably 100 to 150 words as part of this assignment as well. So it's a 250 word max, but if you're under 100, I can guarantee you do not have enough detail for a good abstract section. So that's what's going to be due next week. Again, I'll send out the assignment sheet, but it's going to have a full description. It'll have the data you need to use, have a full description in there. That's worth 15 points. And again, this is your chance to just practice the abstract section before the first full paper. I'll grade those and get those back to you. The other thing to keep in mind is we have our second lab quiz next week. So I handed back your lab quizzes today. Take a look through them. See what you did well, see what you could improve. Okay. Use that as a tool to make sure you're prepared the best for the second lab quiz. But again, the second lab quiz could cover aspects of scientific writing, could cover the last few labs that we've done, including today's lab. Okay. But the first lab quiz is pretty representative of the level of difficulty and the types of questions I'm going to ask also in the second lab quiz. Questions about that? So take a look through. If you got any questions, let me know. But also just make sure you're studying and prepping for that lab quiz next week, because it is kind of like a mini exam. Otherwise, you guys can, abstract assignment. Whoops, I did one. Yeah, 150 word minimum. Before you guys leave today, also turn in your data. So just like last week, I'm going to you guys, once you finish up and find your rates for the 5 drops, 10 drops, and 20 drops yeast concentrations, give me your rates before you leave, and then you're done for the day. Okay. All right, you guys can get started.
Yeah.